So in this video, I'll be talking about the mammalian sex determination. And in mammalian sex determination, unlike Drosophila, the Y chromosome is very essential for the development of the male sex. So if the zygote is XY, it develops into a male, and if the zygote is XX, it develops into a female. Another unique feature about mammalian development is that the gonad develops from a bipotential organ rudiment. So this bipotential means it has the potential to develop into either ovaries or testes. So this is a very unique feature in the development of the gonads. Until 7th week after fertilization, the bipotential gonad cannot be recognized either as males or as females. Now, if XX is present, it leads to the expression of three genes which are important for the female pathway: WNT4, FOXL2, and RSPO1. They allow this gonad to develop into the granulosa and the thecal cells which give rise to ovary follicles. Now these follicles secrete estrogen. Estrogen is a very important hormone. Estrogen also comes from the maternal side. So there is a maternal contribution of estrogen. Now this estrogen, it acts upon the Mullerian duct, which then develops into the female specific organs. On the other hand, if it's XY, then SRY is expressed, SRY and SOX9. These two are very important genes which are expressed, which are required for the male pathway. And the testes or the cells develop into the Sertoli cells and the Leydig cells. Under the influence of SF1, Sertoli cell secretes anti-Mullerian hormone, that's AMH, and the Leydig cell secretes testosterone. Now this anti-Mullerian hormone, it regresses the Mullerian duct so that the Mullerian pathway is stopped, and it leads to the testosterone acts upon the Wolffian duct, which gives rise to the, or which allows the formation of the testicular corns. Now testis also activates a hormone that's called as DHT. If DHT is present, then the male secondary sexual characters are developed. If DHT is absent, then you see a female pathway. So in the Y chromosome, it was found very early that there's a region, if that's missing, then those individuals, even if they have Y chromosome, they don't develop into males. So this was called as testis determining factor. And the testis determining factor, if it's injected into an XX embryo, it gives rise to a female. So these confirm that yes, this region of the Y chromosome is essential for the development of the testes. On the other hand, in the XX, the second X chromosome, the X chromosome is important for the female pathway, but the second X chromosome is necessary for maintaining the ovarian follicles. So if there is just one X chromosome, it develops into females, but the ovarian follicles are not developed and they are rudimentary. Now the genital ridge, which has this bipotential gonad, it expresses genes that's called as WT1 and SF1. SF1, in the beginning, it is present in whether it's a male or a female, but then the level of SF1 sustains if the XY is present, or it's a male pathway. Now SF1 plus other SRY leads to the activation of SOX9. This SOX9 is a very important factor which is present on the autosome, and the SOX9 leads to the formation of expression of testis forming factors and the anti-Mullerian hormone, which we had seen over here. And SOX9 is also important in regulating the levels of FGF9, which is again an important it's important in the male pathway. SOX9, we see over here, it's inhibiting beta catenin. Now what is this beta catenin? Is a transcription regulator which is made by this genital ridge if there is presence of WNT4 and RSPO1. RSPO1, both of them lead to the activation of beta catenin, which is a transcription regulator. Beta catenin inhibits SOX9, and if it's high, it leads to the formation of it leads to the activation of ovary forming genes. Follistatin is important, and follistatin levels are also maintained by another factor that's called as FOXL2. Now this follistatin organizes the granulosa cells, and during development, what happens? XY pathway begins early. If it's not there, then the female pathway starts. Now we look at the different genes that are present, and the different genes that are present are: in ovary we have WNT4 and RSPO1. They both are present in chromosome number 1. Now if this individual is XY, and if there is a duplication of this region of chromosome number 1, then there is sex reversal, and you see the presence of female features. And because of the presence of these two, the beta catenin levels is high. All these genes, they have been experimentally shown to be important in the female pathway. Now, this the Wnt pathway and the RSPO1, they lead to the activation of a factor that's called as TAF2, which is a initiation factor in transcription, and this is seen only in the ovaries. Now, in the testes, we have SRY and SOX9. SOX9 is found in all vertebrates, whereas SRY is found only in the mammals. So, therefore, SOX9 is a more ancestral gene. Now, experiments have shown that the presence of SOX9 is essential for the formation of male. They block beta catenin, and there are several functions of SOX9. It's self-regulation, it blocks beta catenin, it activates a testis-specific gene, it activates the anti-Mullerian hormone, and it's important in activating the FGF9. FGF9 is fibroblast growth factor, and it's very important. What are the functions of FGF9? They are proliferation and differentiation of Sertoli cells, blood vessels from kidney tubules, from kidney and testis. They allow the blood vessels from the kidney, the mesenchyme, to come into the testis. And if you incubate, experiments have shown that if you incubate ovaries with FGF9, you see the blood vessel formation of blood vessels. It maintains the level of SOX9, represses Wnt4, and the most important is coordinates gonad with the germ cells. This is the timing of meiosis. And as you all know that in males, the meiotic events are they do not start until puberty, whereas in females, the follicles, the cells, germ cells, they enter the meiotic pathway and then they are arrested. Another important gene is the steroidogenic factor 1. This is necessary in the bipotential gonad, as we had seen, but if it's a female pathway, the SF1 levels goes down. However, in males it is maintained. This is important. It masculinizes the Sertoli cells and the Leydig cells. So SF1 and SRY activate the SOX9. Now, what you understand is, in development, the timing is more important. The time when the SRY activates the SOX9 gene. Experimentally, if you inhibit or slow down the expression of SOX9, then it takes upon the female specific pathway. So the timing forms a very important feature about males, and so this is about the sex determination in mammals.